Okay, class, so today we're going to cover DNA recombination and repair, and this very much builds off of what we did last time with DNA replication. So DNA repair is essential for normal cell viability, right? If you can't repair uh, changes to the DNA rapidly or any damage to the DNA, it's going to result in a mutation, and then normal cell viability actually becomes compromised. And as we saw last time with replication, the polymerase itself is very high fidelity, right? It has 10 to the fifth normal fidelity, and then when you add the exonuclease activity, it only makes an error 1 in every 10 to the 7th bases. So it's not perfect. And so cells actually need repair mechanisms to really bring it up to the 10 to the 9th fidelity. And so on top of that, you also have random changes to DNA that occur daily, and these need to be repaired, right? If you go to the beach, you're going to get UV damage to your DNA. And also there's a lot of things that we consume that actually can cause damage to our DNA. Carcinogens and genotoxic stress, all of those fall into that. And so ultimately, if it's not repaired, 1 in 1,000 base alterations actually results in a permanent mutation. So we're constantly mutating our genomes over the course of our lives. And the repair enzymes, interestingly, are also used to create diversity and contribute to evolution, as we'll see later on in today's lecture. So here's some inherited syndromes that have defects in DNA repair. Here's what they're called: Bloom syndrome, Werner syndrome, ataxia telangiectasia. Here's a whole bunch of them. Here's the actual resulting phenotype, and here's the processes that they're involved in. And so this brings us to our first homework assignment of the term, where I'm asking you to actually find and describe the mutations associated with the different syndromes and what the proteins do in each. Okay, so in this Werner syndrome, there's an accessory 3' endonuclease and a DNA helicase that can be mutated. And so I just want you to sort of describe what each of one of these do. Use about 3 to 5 sentences for each one. And this homework is due Thursday, February 25th at the normal class time. And you can just send it to me via email. So here are some of the spontaneous alterations that can actually occur to DNA. All right, you can have oxidative damage, and that's just sort of shown with the red arrows here. You can have hydrolytic attack, which is shown in blue, and you can also have uncontrolled methylation in green. Okay, and that can all alter DNA. Now, other things can happen too, where you can get deamination and depurination. And so depurination is when the purines, G and A, actually get hydrolyzed. So these big blue arrows and the actual base sort of gets cleaved off. Deamination is actually when you remove a free amine. Okay, here's one on guanine, cytosine, and up here on adenine. Okay, thymine doesn't get deaminated because it has no amides to basically remove. Okay, so deamination is when basically occurs mostly when cytosine is changed to uracil. There's actually an experimental technique that's used to actually measure DNA methylation, the methyl cytosines, and what you do is you actually treat your DNA with bisulfite in the laboratory, and that will actually convert all of the unmethylated cytosines to uracil, and then you can detect them by sequencing, because the uracil will sort of behave like a thymine in a sequencing reaction. Okay. So here's a closer look at depurination and deamination. So depurination, once again, the base is going to actually be removed. So in this case, guanine basically gets sort of cleaved off, and then you're left with your sugar phosphate backbone. In deamination, in the case here, cytosine, the free amine is going to be removed, and what you're left with is uracil, and the O2 comes in and steals the oxygen. Okay, so depurination occurs basically 1 out of every 5,000 bases daily, so it's a pretty common event. And deamination is even more common and occurs 1 out of 100 bases daily. So these are sort of common occurrences in the cell. And one of the things that I didn't mention here with the oxidative damage, there's a lot of just plain old foodstuffs that can actually lead to increased oxidative stress in our bodies, right? High-fat diet will increase oxidative stress, and that causes, the oxidative damage to the DNA, and that's one of the reasons now scientists are starting to find that there's a correlation between obesity and the obesity epidemic and cancer. Similarly, there's also a strong correlation between alcohol consumption, which when broken down increases reactive oxygen species, which can cause oxidative damage to DNA. And so there's a correlation between heavy alcohol consumption and cancer. Another thing that can happen to DNA is you can actually get UV damage. Okay, so UV light causes pyrimidine and thymidine dimers to occur, and that's basically crosslinks in the DNA, so between these two thymines that are here. Now, in some organisms, there's an enzyme called DNA photolyase that can actually reduce these bonds, and the DNA photolyase is actually circadian and light regulated. But most in mammals and in many eukaryotes and many just animals in general, they don't have DNA photolyase, so these are repaired via a different mechanism, and I'll explain in a bit how that happens. Okay, so what happens if you get any damage to DNA and it's not repaired? Well, ultimately a mutation arises, and it can be a permanent mutation. Okay, so in this instance, if you have a deaminated cytosine, you have uracil, and then when this is actually copied, what's going to happen is if it uses the converted deaminated cytosine, which is a uracil, and it uses that as the template strand, the new strand is actually going to incorporate an adenine in its place, okay, because it can properly base pair with the uracil. Now on the old strand it will be copied normally, but this one will basically leave a mutated copy of the gene around. Okay, if you have a depurinated adenine, right, where the base is actually cleaved off and you're just left with the sugar phosphate backbone, when this gets copied, if this is the copied strand, it just skips the base and you're left with a deletion. Okay, so a single nucleotide is removed from here. And once again, the old strand is fine, and it's copied as it should be, and it remains unchanged. But if these mutations are not repaired prior to replication, a permanent mutation will reside, right? So if the damage is not repaired before the DNA is copied, you'll end up with a permanent mutation in the thing. Some of the DNA repair mechanisms that exist are base excision repair and nucleotide excision repair, and it depends on how much DNA has to be repaired. Okay, if you have just a single base to repair, like in the case with a deaminated cytosine, which results in a uracil, what will happen is the uracil DNA glycosylase will come in, and it'll actually remove the uracil, and then AP endonuclease and phosphodiesterase actually remove the sugar phosphate backbone, and then DNA polymerase can add a new nucleotide, and DNA ligase can seal the nick. Okay, so this is base pair excision. Now, if you have something like a pyrimidine dimer or a thymidine dimer, that's repaired by nucleotide excision repair, where what ends up happening is an excision nuclease comes in and cuts out a big chunk of the DNA. DNA helicase will remove the bad portion of DNA, and then DNA polymerase and ligase come in and sort of fill this out and then seal seal the edges. Okay, so these are two repair processes that I want you guys to know, and you should know when they're used and when they're not used. Now, the next question then becomes, in the case with a deaminated cytosine, for example, or any deaminated base, how does the cell detect that it's bad? Okay, well, it actually flips out because it can no longer properly hydrogen bond, and it's sort of kicked out of the DNA helix, and it will sort of stick out. And so damaged bases are easily detected by DNA glycosylase, and then it'll remove that, and then the base pair, the AP endonuclease, can come in and cleave out that section. What's really kind of neat about DNA is that it has really great chemistry behind it, so that deamination of DNA is sort of easily detected. Okay, so if you have an adenine and it's deaminated, you have what's left over is a hypoxanthine. Okay, and same thing with guanine. If it's deaminated, then you're left with xanthine. Okay, cytosine's a bit unique in that it actually can lead to a uracil, which can be copied. These ones are typically never copied. And thymine actually can't even undergo deamination because there's no amine group associated with it. Okay. And so once again, if you do have deamination, the bases are readily flipped out and can be easily detected because they can no longer form the hydrogen bond with the other complementary base. Okay. Now, one interesting thing is our DNA can be methylated. You can actually get a methyl group added to DNA. In this case, you have the methyl group here, which is in cytosine, and then you have 5-methylcytosine. Now, if 5-methylcytosine becomes deaminated, it actually becomes a thymine, which is a base, and there's no way to tell which is the right base and which was the wrong base. Okay, and this is a real problem because you can have things like there's diseases where this becomes a problem. One example is hepatocellular carcinoma. Okay, so if you have hepatitis C and you've eaten peanuts that have been contaminated with Aspergillus ningulans, Aspergillus ningulans produces this toxin called aflatoxin, right, which actually binds here and looks like it becomes a methylated cytosine. Okay, and when that actually gets deaminated and there's a DNA methyltransferase involved, it actually somehow converts your GC DNA to AT DNA, and that ends up leading to liver cancer. A note on bisulfite treatment. So bisulfite, which we saw before, will actually cause deamination of the cytosines, has little to no effect on 5-methylcytosine, and it only deaminates cytosines. Okay, so that's used in the laboratory to basically detect DNA methylation. And what's interesting about this is that most CpG islands in our genome, they're actually not methylated because if they were methylated like this one, and deamination is such a common occurrence, we would have basically all of the GC-rich regions actually converted to AT-rich regions just by natural evolution. And so most CpG islands are not methylated. Okay. So not only can you have changes to single bases in DNA, but you can also get double-strand breaks and also nicks. Okay, a double-strand break is where both the phosphodiester bonds between the DNA strands are broken, and you get a double-strand break. Okay, and there's two ways to repair a double-strand break. You can have non-homologous end joining, NHEJ. That's an acronym for non-homologous end joining, and then homologous recombination, HR. Okay, or homology-dependent repair. It's also called that. And so if you have an accidental double-strand break, in the case with NHEJ, you get sort of a resection of the DNA and loss of nucleotides, and then these two ends are joined back together. Okay, in homologous recombination, this is a bit different. You get a sort of loss of nucleotides, but then it uses the strands to actually repair it perfectly and no lesions are left behind. Okay, so in the case with non-homologous end joining, you're typically left with a genetic lesion that's detectable, oftentimes a deletion of anywhere from 1 to 4 nucleotides. But homologous recombination is actually perfect and will fix the DNA perfect and fix it flawlessly. And so what's interesting here is the non-homologous end joining, even though it's error prone, is actually the more preferred method of repair in a mammalian cell. Okay, and in a somatic cell lineage, one can actually detect somewhere close to 2,000 different NHEJ repairs over a 70-year lifespan in a somatic cell. Okay, so we're getting these NHEJ lesions, throughout our body as we age. Here's how NHEJ works. So non-homologous end joining, you'll get a double-strand break. And then this double-strand break is recognized by these enzymes Q70 and Q80 that bind to both ends of the breaks. The histone H2A in the chromatin is actually replaced with the histone variant gamma H2AX. And then this recruits other factors called XRCCs, which sort of then process the DNA ends and gives a little limited repair synthesis. And then this enzyme called Lig4 actually ligates the ends back together and seals the nick. Okay, so generally there's a small deletion that occurs through anywhere from 1 nucleotides to 4 nucleotides. So this is a cellular repair process that I want you guys to know, plus the base pair excision and the nucleotide excision repair along with it. And here is basically the structure of Q70 and Q80 bound to DNA. Here you can see the double-stranded helix where it's been sort of broke and it's sort of looking down the DNA. And the Q70 and Q80 are shaded different colors green. One important thing about Q70 and Q80, these are actually also involved in VDJ recombination, which is a really, really cool subject matter, which those of you who've taken immunology will know about. So during hematopoiesis, and hematopoiesis is the process basically where a hematopoietic stem cell in our bone marrow gives rise to all of the different cells in our blood, right? White blood cells, red blood cells, macrophages, neutrophils, platelets and whatnot. So in the case with VDJ recombination, it actually happens in B cells and T cells, mainly in B cells, where you get sort of a genetic recombination of the antibody sequence in a B cell, so that every B cell that sort of is released into our bloodstream produces a somewhat different antibody. Okay, and then those antibodies then, once they detect a specific antigen, they can be amplified. And that's what's happening when people are getting their COVID-19 vaccine. They're basically selecting for specific B cells that recognize the spike protein on the coronavirus. And then they're having a good antibody response because those B cells will actually undergo what's called clonal expansion, and so you'll get many of the same B cells producing the same antibodies against the virus. Okay, now on to CRISPR-Cas9, because I'm sure you guys have all had this in your class. It's at some point in time. It's sort of the latest and greatest genetic technique that came out about 10 years ago. And so the way it works is you have this nuclease called Cas9, and then you have a guide RNA that can complementary base pair to a specific sequence, and then based on where the guide RNA is actually associating with DNA, that guides the Cas9 nuclease to create a double-strand break. Okay, and normally this double-strand break that's produced is actually repaired by non-homologous end joining, so it will resect the DNA and you'll be left with a couple of mutations. So it's serving right now in the research community as a very, very powerful technique to create directed genetic lesions to study gene function. But the long-term goal and the hope of the CRISPR-Cas9 system is in the ability to actually have it used for gene therapy, where you could create a targeted cut at a gene that's non-functional and theoretically put in a piece of donor DNA that can actually repair the genetic mutation. Now, one of the important things about knowing homologous recombination and non-homologous end joining is that currently non-homologous end joining is the preferred mechanism of repair for this, so it would just leave behind another mutation. However, the goal of this system is actually to be able to introduce a good copy of DNA that can be repaired by homologous recombination, and then that can actually be used for gene therapy. So CRISPR-Cas9 has been used in a lot of different systems, from human cells to zebrafish to bacterial cells. There's really no limit on the CRISPR-Cas9 system. And so it really offers great promise. Okay, now on to homologous recombination, because homologous recombination is important in a number of processes, and I really want you guys to appreciate and know a lot about homologous recombination. Okay, one area where it's used a lot is during DNA replication. Okay, so DNA nicks can be quite common, and that's a single cut in a single strand of DNA. Okay, so as the polymerase is actually moving down and creating the replication fork, if it comes across a DNA nick, it's going to basically break and the fork breaks. Okay, and so then you're basically left with what looks like a double strand break, this is sort of catastrophic for a cell and it needs to be repaired. Okay, and so an endonuclease can come in and it actually degrades the 5' end of the DNA. And this basically creates sort of a 3' overhang that can now undergo a strand invasion of the old strand. Okay, and then synthesis can actually start to proceed, and there's a strand breakage and religation, and everything can proceed, and then the replication fork can start again, and then the Okazaki fragments on the lagging strand can be created, and all this back stitching can occur. Okay, so this is what happens when the DNA polymerase actually hits a nick in DNA. And once again, this is a process that you guys should all know how it works to repair in the case that the DNA polymerase hits a nick in DNA, because it happens quite a bit. Now, in order for homologous recombination to occur, you actually have to have complementary base pairing. Okay, so here's just a schematic of some non-pairing interactions, and then you start to get some pairing, and then that will lead to a sort of a helix nucleation event, and then you'll get basically rapid zippering. Okay, so this complementary base pairing is required between the invading strand and the homologous strand. And this actually creates what's called a heteroduplex. Okay, and we'll see the heteroduplex over and over in the next couple of slides. Now, in order for homologous recombination to occur, you have to have this enzyme called RecA. It's Rad51 in mammals, RecA in bacteria. Okay, and this binds to the 3' end of the single-stranded DNA to help the strand invasion. Okay, so this little portion that's right here, it's going to bind to that and then help the strand invasion step. Okay, and here's basically just the sort of space-filling model of it, and here you can see the different subunits here, or the three copies of it bound to DNA. Okay, and so what RecA does is actually help form heteroduplex DNA. Okay, and it starts with this sort of 3-stranded structure, and then you have your heteroduplex DNA, and this actually uses ATP to basically create the heteroduplex, and then that helps to allow for branch migration of this down so that you can get a rather large size of the heteroduplex DNA. HR in eukaryotes actually requires this other enzyme, RAD52. And so what RAD52 does is it displaces single-stranded DNA binding proteins, right? We saw those single-stranded DNA binding proteins during the DNA replication, right? So if you have a piece of single-stranded DNA, the single-stranded DNA binding proteins are going to want to bind, but you actually need to put RAD51 on for the strand invasion. So RAD52 actually displaces that, and then that assists with the strand invasion and eventual branch migration. And the branch migration is actually fairly important, right? And to have the branch migration regulated is an important aspect of all of this. Okay, so normally if you got your heteroduplex DNA and you have this branch point, it can sort of go back and forth. There's nothing that's sort of preventing it from going in either direction here. So in the case with RAD51 and RAD52 and some proteins I'm going to show you in a little bit, the way this works is you can sort of get branch migration, and it's directional. Okay, so you can sort of control the amount of heteroduplex DNA that happens in this whole process. Okay, so you want sort of protein-directed branch migration, so you have a substantial amount of the heteroduplex DNA. Okay, so here's how homologous recombination works when you get a double-strand break. This is a very sort of simplified schematic. You get your double-strand break. An exonuclease comes in and basically chews back in the 5' to 3' direction, leaving you with 3' overhangs. RAD51 and RAD52 will then bind to these 3' ends and actually cause a strand invasion. The strand invasion actually happens on both the strands of DNA, not just one. And then what ends up happening is you get the branch migration, and then all of a sudden the DNA polymerase can come in and start to polymerize what's needed, and then you'll get a breakage, and basically the strands are basically ligated back together. And so this green section of DNA actually used the red DNA as a template to be copied to fix everything, right? Because you're sort of chewing back stuff at the end. Okay, I hope that makes sense. We'll see homologous recombination again in a bit, but here's how prevalent it can occur in a cell. Okay, so in the case with double-strand breaks, they're rapidly repaired. So this is just a DAPI-stained image of a nucleus. It's stained in blue. And if you hit this cell with strips with X-rays to basically generate double-strand breaks, and then you add a thymine analog called BRDU to the cell, which is labeled green here, it will start to be incorporated. And then if you look at MRE11, which is the exonuclease involved in sort of chewing back the in the 5' to 3' direction on both DNA strands, that's shown here in red. Okay, and ultimately when it's repaired, what you end up getting temporarily is this thing called a Holliday Junction. Okay, and a Holliday Junction is a 4-stranded intermediate that's produced when 2 of the double helices sort of come together. Okay, and proteins that are involved in helping to stabilize this Holliday Junction are called RuvA and RuvB. And here is what RuvA and RuvB look like in this nice, beautiful model. You have RuvA here in green, and it's a tetramer, and then you have RuvB, which helps move the DNA out, and that sort of pulls the DNA in, and so you can have that branch migration. Okay, and you can sort of think of RuvB here. It looks very similar to a DNA helicase, and it uses ATP to basically slide the junction around. Okay. Now, homologous recombination is actually a regulated event that occurs during meiosis. And this is actually pretty cool because it allows all of you to have basically DNA from both your grandparents on one chromosome, right? And so the way it works is when you have a diploid cell, so when the germ cells are actually produced, there's a replication event, and then they undergo meiosis. And during this whole process, you have sites of gene conversion where you can have homologous recombination producing heteroduplex pieces of DNA as well as crossover events. Okay, so these two chromosomes right here can become mixed and matched. Okay, and so for example, in your mother, she will have one chromosome from her father and one chromosome from her mother, and then by the time the germ cells are passed on to you, your grandparents' chromosomes are all mixed and matched to become your chromosomes. And like I said, this is actually a regulated event to control genetic diversity, and it's controlled by this enzyme called SPO11, which actually creates the double strand breaks. Okay, so the double strand breaks that are occurring here are actually a regulated event. SPO11 will come in and cleave them. And so this is how it works. And once again, this is something I really want you guys to know. So what you'll have is your paired homologous chromosomes. SPO11 comes in and will create a double strand break. MRE11 is a nuclease that will chew back in the 5' to 3' direction, and then RAD51 and RAD52 will bind to the 3' overhangs here, and that will facilitate the strand invasion. And then you will get the DNA polymerase to come in, and you'll start to get polymerization and copying of the two strands, and eventually these will be sealed together by DNA ligase, creating your double Holliday Junction. Okay, and then when you have that double Holliday Junction, which is sort of stabilized by RUV-A and RUV-B and sliding along, at some point in time there can be a cut and a religation. Okay, and depending on where the cuts occur, it's going to determine whether or not you have just plain old heteroduplex DNA, or if there's a crossover event that has occurred. So if the cut occurs here, what you're left with is heteroduplex DNA without a crossover. If the cut occurs here, you'll basically have, and a cut occurs here, you'll have basically heteroduplex DNA that also contains a crossover event. Here's just a closer look at it. So once again, you're going to have after strand invasion, the DNA polymerase is going to start to synthesize the DNA. One important thing here for you guys to appreciate is once again the directionality of DNA replication and what it's using as a template strand. Okay, so the polymerase is extending bases in the 5' to 3' direction, copying the DNA off the 3' to 5' direction. Okay, so once again, this is 5' to 3', and it's extending along, and it's basically traveling copying the DNA from the 5' to 3' direction. So once again, in order for you guys to really appreciate homologous recombination, you have to know how the DNA is being copied, okay, on this strand, and you want to make sure that you have your DNAs properly aligned as is shown here, right? So the directionality is always going to be 5' to 3' and 3' to 5', okay, with DNA. Okay, so once you have your double Holliday junctions that are formed, once again, the cut, if it occurs here and occurs here, what you're going to get is just a heteroduplex without a crossover. If it happens here and it happens here, across here, you're basically going to get heteroduplex DNA with a crossover. Okay, and so here's what they're going to look like. Here's just your plain old heteroduplex without a crossover, and then your heteroduplex with a crossover. Now, homologous recombination actually can cause gene conversion too. So if you have basically a heteroduplex that forms in a gene, and there's different alleles and different DNA sequence in there. What will ultimately happen is you'll get base pair excision, or the nucleotide excision, where you'll basically remove a whole portion of the DNA, and then one strand will get copied and repaired any of these sort of mismatches. And this is random. It can come from either strand. Here it's shown in red, but it could have equally fixed the blue and carried the blue out. And this is one of the culprits of sort of gene and domain duplications that we saw early on in the term when we were talking about DNA and how, you know, enzymes evolved, and how certain enzymes have the same protein domains within them, okay, with different domain folds with oftentimes different catalytic activity. So homologous recombination is tightly controlled. And so here's basically an example of that. Here you have similar but not identical repeated sequences. Occasionally you can get sort of the strand exchange, and then if they detect these sort of mismatches, everything's aborted and it goes back to normal. But if that proofreading fails, a couple of recombination events can happen where you basically get a crossover and you get a big loss of DNA, or you can have it where you get the heteroduplex DNA and changes are further incorporated. And it turns out there's a ton of homologous DNA in mammals, and that's one of the reasons that homologous recombination is relatively rare, and non-homologous end joining is the more favored DNA repair mechanism in the case with a double strand break. Okay, now we're going to talk a little bit about transposable elements, because a lot of our DNA in our genome is actually former transposable elements. Okay, and these basically occur in almost every organism imaginable. Okay, and so what you end up having is a transposase with oftentimes short inverted repeats, and so here's an example. They occur in Drosophila, in plants, E. coli, in this organism Snapdragon, which I actually don't know what it is, and then there's basically retroviral-like transposons that occur in a lot of different organisms. One is TAG in humans, and then there's also non-retroviral retrotransposons like L1 in humans. Okay, so these are actually very common, and they make up a large portion of our genome. Here's some of the common bacterial transposons: IS3, TN3, TN10. The important thing for you guys to know is that they have these inverted repeats on them. Okay, and that actually helps. And these are very powerful genetic elements in bacteria. So if any of you guys go on to study microbiology, you might come across these in your studies. But here's how they work. You can have a transposon in chromosome A, and then the transposase can sort of find the inverted repeats. These are oftentimes short inverted repeats, and what they can do is sort of lop themselves out of the chromosome, and then the broken chromosome is then repaired. And then you have this sort of mobile genetic element now, the transposon, which can actually reintegrate into a target chromosome, and part of it is because of these inverted repeats. Okay, so here's the transposase structure. Here you can see it bound to the inverted repeats on the DNA. Okay, and this is the transposase. Now, the reason this is sort of important is because certain human viruses and just animal viruses in general can use transposition. And a perfect example is HIV. Okay, so HIV is an RNA virus that contains an enzyme called a reverse transcriptase. And other viruses have this as well, but I'm sort of just going to go through it with HIV. Okay, so you'll have your RNA in the capsid and envelope, and that can infect a cell. And then what's going to end up happening is the reverse transcriptase is going to copy the RNA into a DNA strand. Okay, and then that DNA strand will then be copied into double-stranded DNA, and that can actually integrate into the genome. Okay, and this is one of the big problems with HIV. So if you've ever had an HIV infection, you have the HIV sequence in your genome for your entire life. And so you will have to be on whatever drugs suppress the HIV from basically growing and replicating in your cells. Okay, and so what ends up happening once the DNA is integrated, the RNA is actually expressed, and the RNA from the virus can make the capsid protein, the envelope protein, all the other things that the virus needs, and you'll get basically production of more viral particles. Okay, all containing the RNA and the reverse transcriptase, and then they can go on to infect other cells. And so this is just an example of a retrovirus, which DNA is, and so it'll incorporate into the genome, and then you're sort of stuck with it the rest of your life. The enzyme here that copies the RNA to DNA is called reverse transcriptase. Here is actually the structure of reverse transcriptase, and it takes basically a template RNA strand and it converts it into a DNA strand. Now, interestingly, we use reverse transcriptase in the lab a lot to convert RNA to DNA. It's a common enzyme. You can buy it from a bunch of biotech companies if you need to use it. And it's used for experiments like real-time RT-PCR. And actually, it's a common reagent now used to actually detect the coronavirus, because the coronavirus is an RNA virus, and to basically detect in all of these sort of saliva tests and nasal swab tests, what they do is they sort of try to get a collection of the coronavirus. They then isolate the coronavirus RNA. They use reverse transcriptase to convert it into DNA, and then they can do quantitative PCR to see if the DNA, which was copied from the viral RNA, is actually present in that sample. And that's how a lot of the coronavirus testing is occurring. Okay, so here's how viruses integrate into the genome. So you'll have your viral DNA. The integrase will sort of process the viral ends, and then you'll basically have an attack of the viral DNA onto your chromosomal DNA. Then there's sort of gap-filling repair, and it's integrated into the genome. Okay, and here's basically how the integrase sort of helps to catalyze that reaction. The free hydroxyl groups can come in and attack the DNA. Now, in the case with non-viral transposons, which are sort of present throughout our genome, this is using L1. So L1 will be on our chromosome. It'll actually synthesize L1 RNA. Reverse transcriptase and endonuclease combined to it, and then you'll get a cleavage of the strand, and then that actually serves as a primer for the reverse transcriptase to fill it out, and then in a multi-step process, that's just copied, and you get a whole new L1 element in a different region of your genome. Okay, so if you sort of look back at sort of an ancient human genome, millions and millions of years ago, we might have had one copy, and then all of a sudden it was basically expanded to the point where we now have basically a million copies of this ALU sequence, which is sort of one of these non-viral transposons that has sort of jumped in all over our genome. Here's just a rough schematic of how integration, excision, and inversion can occur. You can see you have a circular piece of DNA here, and it can find a region to sort of pair with, and then it can sort of integrate in, and then actually this can also be excised out, going back to the other way, and you can have an inversion where sort of A and B come together, these repeats, and then it can sort of flip around, and then you're left with B to A. So all these kinds of different genetic events are happening all the time in our genomes, just to give you guys some idea about how dynamic things are in our DNA. Now, in the case, here Here's Lambda Phage. Those of you guys who are interested in microbiology should really pay attention to this. I'm sure you've had it if you've taken Professor Boyd's microbiology course. And so what you have here is Lambda Phage. It's a circular DNA phage, and what it'll do is it has a lambda integrase protein that'll bring it to a region of the genome, and then it can basically create a double strand break and join, and then the lambda phage DNA is actually integrated into the bacterial genome. Here's basically the life cycle of lambda phage. So you'll have an E. coli strain, and you get the lambda phage. It'll inject its DNA. It's circular. It will then get integrated into the E. coli chromosomal DNA, and the cells will just continually divide, and this is called the prophage pathway, where the phage DNA is actually just incorporated into the E. coli genomic DNA, and it can replicate and replicate. But every now and then there's an induction event where the DNA gets kicked out of the genome, and all of a sudden it will start to replicate, and all of the phage proteins will be made. Eventually the DNA is packaged into the phage and the cell lyses, and that's called the lytic pathway. Now, similar genetic techniques are also used in mammals, where we can actually integrate different things in and then create sort of experimental systems. And one really great system, especially for like doing mouse genetic work and other things like that, is the use of this system called LOX-CRE. Okay, so CRE is a recombinase gene, and oftentimes you can put it in a specific tissue under a tissue-specific promoter. Okay, so it's integrated in sort of mouse ES cells, and then you can create a transgenic mouse line with this CRE recombinase under the promoter over a tissue-specific promoter. Okay, so in this case, if it was a liver-specific gene or a liver-specific promoter, the CRE recombinase would be produced, the mRNA would be expressed, and you would get CRE recombinase made in liver cells. Okay, you can cross that to a mouse that actually has LOX-P sites around a gene of interest. Okay, and so the LOX-P sites are actually where the CRE recombinase will sort of excise a piece of DNA. Okay, so the CRE recombinase will recognize these LOX-P sites, and in the case with liver tissue, if this gene is in the liver, these LOX-P sites will be removed because the CRE recombinase is under the control of a tissue-specific promoter, and it will pop it out. And this way you can actually make tissue-specific knockouts of important genes to understand various cellular processes. So this is used widely in the biomedical research field. In other tissues, the gene of interest is not expressed. So for example, in muscle cells, if this is a tissue-specific promoter, like for liver, if this is a muscle cell, the gene will not be expressed, and then the normal gene of interest is there and it is made. And so this allows you to create tissue-specific knockouts. This is really important for all the graduate students to know how this system works. I'm not going to hold the undergrads to knowing this, but grad students, you guys need to understand this because it's a really important genetic technique that's used all of the time in biomedical research. And I feel it's that important for you guys to know. Okay, so that's all I have for today. So next time we'll talk about transcription and splicing. The reading are basically chapter 6. And just a heads up, the next exam or the first exam 1, will be next Thursday, February 18th. At the normal class time. And the exam will cover everything we've covered up through Lecture 9. So that will include transcription and splicing and translation, DNA repair, DNA replication, and a lot about the histone. So it's a pretty, it's going to be a pretty packed exam. So if you're a little bit behind with the course, now is a great time to catch up. Okay, so I will see you guys all later, and I hope you enjoyed this lecture. Bye-bye.